In this lecture and the coming lecture, we'll focus on the enzymes, especially their structures, their types, as well as the mechanisms for their enzymatic catalysis. So far, we have studied the macromolecules and their properties, such as nucleic acids, sugars, and polypeptides. Now, let's switch to their inner conversions, especially in the context of biochemical reactions, to better understand how they are catalyzed by enzymes and their mechanisms, as well as the rates of such inner conversions and the regulation for them. So there are two big sisters for better understanding these enzymes' activity as well as biochemical reactions. The first one is structural biology, which is the study of the structure of macromolecules and their interactions. So that basically helps us understand how these enzymes and the substrates work in 3D in terms of their structures on the interface, as well as some functional domains and the atom alignments and so on. And the other sister is kinetics, which is the study of reaction rates. So rather than focusing on the structures themselves, it focuses on each steps of these reactions and how fast or slowly they're happening, and also how these dynamics can be controlled and regulated. And both these structural biology and kinetics contribute to understanding of the reaction mechanisms, such as reaction steps, participants of such biochemical reactions, and the functional group movements, as well as actual chemistry in the enzyme's active site. And combined use of these structural biology and kinetics is fundamental to rational drug design too, which focuses on any kinds of enzymatic reactions that specifically working on the therapeutic targets. Again, enzymes are the biological catalysts. Now let's talk more about the catalysts in general, focusing on their properties. First, a catalyst is something that speeds up the rate of a reaction, and they're not going to be consumed or altered in the reaction, meaning that they are not the reactants or the products. And the catalysts, including enzymes and the biological versions, do not change KEQ nor delta G naught of the process. And I hope you'll still remember what these state functions are, which we learned in the earlier of this course in the thermodynamics section. Briefly, KEQ, equilibrium constant, is the ratio of the product and the reactants in their concentration. And delta G naught is the Gibbs free energy that determines or shows the spontaneity of the reaction. The catalyst just influences how quickly the equilibrium is achieved. Again, it only affects the speed or the rate of the reaction. And remember, the KEQ is related to delta G naught by this equation that we've already learned in thermodynamics. And that means that enzymes can only affect the kinetic properties, but not the thermodynamic properties of the process. Again, catalysts cannot or do not change the KEQ or the delta G naught. An example of the catalyst will be the oxidation of the glucose, where you can see the reaction here with a pretty high exergonic process. So you can see a lot of energy that is generated from this reaction, which means that the delta G naught, the Gibbs free energy, is highly negative. So this reaction itself is highly exergonic, again, generates a lot of energy, but that does not occur outside of the living cell with appreciable rate. So its rate is really slow. Therefore, the large negative value for the delta G naught does not mean that the process is fast, but it only tells the spontaneity of that the reaction. Again, it does not have anything with the rate of the reaction. This is the reaction diagram for uncatalyzed reaction, where this red curve shows the energy profile as the function of reaction progress. So A and B are the reactants of the reaction, and the P and Q are the products. And obviously you can see there's a delta G, the difference in the energy between the reactants and the products indicated here. And this is a delta G with the negative values because products have the lower energy. So it would have been a spontaneous process if there wasn't this huge hurdle in terms of energy changes. And this top point, which is the highest energy within this energy profile, is called as a transition state, which is a highly reactive, high energy state. So in order for these reactants to become the products, it actually has to go up to this transition state where it requires a lot of energy input. Then naturally it'll go down to this product where there's a low energy state. But this reaction still occurs because a certain fraction of the molecules of the reactants, which are A and B, has large enough energy to achieve this transition state. And the difference between, in terms of the energy difference between this reactant and the transition state is indicated here as a delta G double dagger, which shows the free energy of activation, or delta G of activation. And depending on the size of this delta G double dagger, it is this reaction from A plus B to P plus Q can be easily achieved, or have a really hard time to get there because of a huge hurdle. The rate of a reaction, or the reaction speed, is proportional to the fraction of the reactant molecules having enough energy to achieve the transition state. So the more the reactant molecules achieve the transition state, the more product that you'll get. And that fraction is described by the Maxwell-Boltzmann distribution, which is shown here on the graph of the fraction of the molecules versus the energy that they have. So the molecules that are distributed or located here on this region, which have a relatively high kinetic energy, will have more energy and easily get to this transition state, as opposed to the molecules that are distributed on this region with the low kinetic energy will not have enough energy to overcome this transition state or get to the transition state, so they're gonna just stay at this A plus B state, so just their reactants molecules themselves. So and also this Maxwell-Boltzmann distribution changes depending on the temperature. So for example, in higher temperature, it shifts to the rightward, so it'll naturally have more bigger population that will have the same or the above a certain kinetic energies. So in higher temperature, normally you can think that there are more of the fraction of the reactants that can move to the transition state for the further process for this reaction. So the higher the delta G double dagger, or the free energy of activation, the smaller the fraction of the molecules that is capable to achieve the transition state. And in the opposite way, if this delta G double dagger is small enough, then they're going to be larger fraction that can easily get to the transition state. And this process, basically, that moving from the A plus B to P plus Q through the Maxwell-Boltzmann distribution is described by the Arrhenius equation, where you can see the equation right here. K, which is the rate of the reaction, equals the A, which is constant, that is specific for a given reaction for this Arrhenius equation, multiplied by the E to the power of the negative or the minus delta G double dagger divided by the RT. And you can move this minus value in the exponential function here outside to get this A divided by E with the same power here. And again, this K, the rate constant of the reaction, is a proportionality coefficient in the rate law. Again, here's the Arrhenius equation, so we don't need to go back to previous slide if we need to look at it. And let's say that there's a process of conversion of S into P, where S stands for the substrates and the P for the products. The rate of the reaction V will be the product of K, the rate constant of the reaction from the Arrhenius equation, and the concentration of the substrate. And when you look at the Arrhenius equation itself, delta G double dagger and the K has an inverse relationship, meaning that the bigger the delta G double dagger, the lower the K is. And of course, the K has the correlation with the rate V. So if the K decreases, the rate also will decrease. So you're going to have a slower reaction. And there are several ways to speed up the reaction rate V, where you can change the K value or the concentration of S. Let's focus on this K value. When you raise the temperature, which is right here, even if it's in the denominator, if it is increased, then because of this exponential function with the negative values, you're going to get K that is also increased. So you're going to get higher, larger K, meaning that you're going to get the faster reaction rate. And you can do the same thing basically by lowering the free energy of the activation here. So again, this is an exponential function with the negative value right here. So if you lower the this free energy of the activation, you're gonna get the also higher K. So it'll speed up the reaction again. And for many chemical reactions, rate increases 2 times by increasing temperature about by 10 degrees C. And raising the temperature raises the energy of the reactants. Again, if you have the more fractions, higher fractions with the high energies, then it will help you to get to the speed up the reaction. And the catalyst decrease delta G double dagger. So here's the uncatalyzed reaction curve, which is in blue. But when the enzymes or the catalysts are involved, that lowers this delta G of activation value, but without changing this energy state for the reactants or the products, it will boost this rate of the reaction. And the difference between the old one, uncatalyzed reaction delta G double dagger, and the catalyzed delta G double dagger would be delta G double dagger, and sometimes you put the cat for catalysis here too, but it can be omitted. So both this 1 and 2, raising the temperature and lowering the free energy of activation, lead to an increase in the fraction of molecules, especially the reactant molecules, possessing sufficient energy to achieve the transition state. So it'll speed up the reaction. The degree of rate enhancement by an enzyme is related to delta G double dagger, or the differences in free energy of the activation between the catalyzed reaction and uncatalyzed reaction. So from the Arrhenius equation, where K can be written as this, uncatalyzed reaction, the K1 will be written as this with the delta G1 double dagger. And you can see it right here too. Delta G1 double dagger will be indicating this much of energy changes for the transition state. And catalyzed reaction definitely will have smaller with the delta G2 double dagger for their energy for transition state. So it can be written as this. And then degree of rate enhancement is basically the ratio of rate constant. So you can get it from the K2 divided by K1, where you can get them here. And when you plug these equations into that, and since these are the exponential functions, by following the rules handling these functions, you can actually change the denominator and numerator here to get it. And then by dividing the same values, you can actually use the subtraction between these exponential values. So ended up getting E delta G1 double dagger minus delta G2 double dagger, which are together divided by RT. And this difference from the delta G1 double dagger and the delta G2 double dagger would be delta G double dagger. Again, differences in free energy of activation between catalyzed and uncatalyzed reaction. So it can be finally written as this here, where the again delta G double dagger is the difference between catalyzed and uncatalyzed one, and also can be written as this with the cat for catalyst or catalysis. And since this is the exponential function, delta G double dagger of just 5.7 kilojoule per mole, which is smaller than one hydrogen bond, gives tenfold rate enhancement. And even 34 kilojoule per mole, which is about 2 hydrogen bonds, will give 10 to the 6th rate enhancement. So that's going to be a huge changes. Before we talk more about the catalysis with more complicated reactions, let's consider a simple reaction where it goes from A to B. And an example that I'm showing you here is a carbohydrate and that is altering its conformation to a more energetically favored chair conformation here from a boat conformation. And during the reaction, the molecule reaches a point of highest energy corresponding to a half chair here, as you can see here, which is the most strained and flattened state, and it has the high energy state of the structure between this boat and chair conformations. And of course it has the highest energy state, and this point where it has the half chair would be the transition state that goes conformational change basically from A to B. And you can still define the delta G double dagger as well as delta G naught too for such reaction. And typically we think that this rise of the energy or the transition state is needed to ensure that the reaction doesn't always occur, but with some kind of control or the energy input, you can actually have more control on this reaction, even though it's a spontaneous reaction. So far, we've talked about the general catalysts as well as how they speed up the reactions. Now let's focus on the enzymes, which are the biological catalysts. They also accelerate the reaction rates in the cell or in the living organism via lowering delta G double dagger or the free energy of activation. And they work via combining transiently with the reactants and promoting their entry into transition state, just like catalysts. But enzymes are the best catalysts on Earth. They differ from chemical catalysts in several ways, and I've listed in this 5 different ways that the enzymes are acting differently. And the first one is the majority of enzymes are proteins, although some are RNAs, as opposed to many chemical catalysts are just acids, bases, and metals. And enzymes are typically more efficient, so they allow higher reaction rates as 10 to the 6th to 10 to the 12th power, higher than the uncatalyzed reaction, and still 10 to the 4th and to 10 to the 8th higher than the chemical catalysts do. And they work under mild conditions such as 37 degrees C, which is a body temperature, 1 atmosphere, and at the neutral pH, as opposed to chemical catalysis works under extreme conditions. And they have a greater specificity to the substrate. And substrates are basically the substances that enzymes work on. And also they're specific to the reaction type too. So for an example, they have a stereospecificity in terms of their substrates, which is why most amino acids in living things are L form of amino acid, and sugars are in D conformation. And that's because of this stereospecificity of the enzymes that are working there, and only these enzymes recognize that specific forms for that, and these are the only types of enzymes that are expressed in the living system. And there are no non-productive side reactions. And typically, one enzymes work with the one substrate. And enzymes also can be finely regulated by altering the enzyme level through the changing the synthesis and degradation rate of this enzyme expression, because enzyme is again the proteins that are expressed from the genes. So at the DNA level or the RNA level, the living system can regulate this enzyme level. And also, this enzyme can be regulated by the changing their activity through the covalent modifications of enzymes, various classes of inhibitors and activators, including allosteric players. And enzymes reduce the free energy of activation delta G double dagger through binding to substrate or S to form the ES complex, enzyme substrate complex, as you can see here. And binding occurs in the active site of the enzyme, and that's the same site where the actual chemistry takes place for this catalysis. And binding takes place via non-covalent weak forces, and that is the same type of forces that hold together the protein structure too, such as ion or electrostatic interactions, hydrogen bonds, van der Waals interactions, and hydrophobic interactions too. And usually the enzyme has a distinct cleft into which the substrate fits. So it has a distinct shape there, right here, the clefts that perfectly fits with the substrate. So about the fitting of the substrate into the enzyme, there are two aspects of such complementarity: geometric and electronic complementarity. Geometric complementarity, as it says, is about the knobs on the substrate that fits really well into the holes on the enzyme, or vice versa. And electronic complementarity will be about the attractive interactions between the substrate and the enzymes because of the electronegativity and the electropositivity interactions. Then if enzyme binds a substrate very tightly, would that mean that it is an efficient enzyme? It's not going to be so simple to answer, because the enzyme must bind well with the substrate in the transition state, but not just a substrate binding very tightly anytime. Because if that's the case, then it will make a hard time to get to the transition state. The role of an enzyme is to lower delta G double dagger, or the free energy of activation, and this is usually done by stabilizing the transition state. And there are two models that have been proposed to explain how these enzymes work. The first one is the lock and key hypothesis that was proposed by M.O. Fisher in 1894, and it explains well about the substrate specificity, where each of them work as lock and key, literally there. But it says nothing about why catalysis occurs, because it's all about the lowering the energy of the activation, but it can't really explain anything about that. There was another model called the induced fit hypothesis that was proposed by Dan Koshland in 1958, and that, in there he proposed that enzymes prefer to bind to a distortion of the substrate that resembles the transition state, and both enzyme and substrate must adjust to one another. So they're going to have a sort of a conformational change to get to the transition state and accelerate this reaction. But in reality, enzyme is not distorted, but has evolved to bind in a certain way and sometimes undergo conformational changes to best fit the transition state. And we're going to discuss more about this with an example in the following slides. An example of induced conformational change during the reaction will be the hexokinase. You can see its structure here with the D-glucose before and after binding for the reaction. And this hexokinase catalyzes phosphorylation of the glucose into glucose 6-phosphate during glycolysis, which is the process of digesting the sugars. And it's pretty unusual enzyme because it has the huge conformational changes. But not all enzymes undergo such a large conformational change. But not only enzymes change their conformation, but many structural and motor proteins undergo large conformational changes during their cycles too to form the higher order structures. So that mechanism definitely is used not only for the reaction or the substrate process, but for the making some kind of scaffold or the structure, or even for moving the muscles. High degree of enzyme specificity largely due to lock and key principle. So again, remember the stereospecificity where it has the handedness basically. But and the degree of geometric specificity varies depending on their structures of the active sites as well as the substrates that they're binding. But often enzymes will act on a group of similar substances rather than just one particular substance. As long as they have a similar structure or some kind of similar features in terms of their interactions, they're going to actually work on them too, regardless of different efficiencies. And some are fairly promiscuous. So for example, chymotrypsin cleaves the peptide bonds at C-terminal region to any bulky hydrophobic residues. And even it can also cleave the ester bonds too. Enzymes are named in several different ways, so nomenclature of the enzymes would be followed by trivial rules like a catalase. You can just name them because that has been called that way for a long time. And the systematic way to make the substrate name plus A's there. With the classification number assigned by the IUBNB. So the example would be 3.4.3, 23.3, things like that for catalase. And typically add this A's to the name of a substrate. So an example is lactase, which breaks down the lactose. And again, I hope you remember lactose is the disaccharide of glucose and galactose. And IUBNB classifies enzymes based upon the class of organic chemical reaction catalyzed. So here are the classifications. Here, oxidoreductases are basically the enzymes that are involved in redox or the oxidation reduction reactions. And transferases is for the transferring some groups, the functional groups. Hydrolases are for the hydrolysis reactions, and lyases for breaking down some bonds like double bonds here. Isomerases are for isomerizing the substrates, and ligases are for the bond formation using the ATP hydrolysis. So far we talked about different types of enzymes. Now let's discuss in depth of their mechanisms. And a good example would be the serine proteases, which have been extensively studied. And as serine proteases, or also called as serine endopeptidases, are a class of peptidases that are characterized by the presence of a serine residue, especially serine 195 residue, in the active site of the enzyme. And serine proteases are grouped into clans that share structural homology, and then further subgrouped into families that share close sequence homology. And the major clans found in humans include the chymotrypsin-like and subtilisin-like and alpha-beta hydrolase and signal peptidase clans. And serine proteases participate in a wide range of functions in the body, and it is really critical in a lot of biological processes, including blood clotting, immunity, and just inflammation, as well as contributing to the digestive enzymes in both prokaryotes and eukaryotes. Among serine proteases, chymotrypsin, trypsin, and elastase are the three chymotrypsin-like serine proteases that have been studied in the greatest detail. And what you're seeing here are the chymotrypsin and the bovine trypsin structures, where they indicated the critical residue, serine 195 here and here in the active sites of those enzymes. And all three are synthesized by the pancreatic acidic cells and secreted in the small intestine for the digestive process. And all three of these enzymes are similar in structure, which was shown by X-ray crystallography. And the differing aspects between these enzymes lies in the peptide bond which is being cleaved, which is a cis bond. So among those peptide bonds, the one that is targeted by these enzymes are called cis bond. Each of these digestive serine proteases targets different regions of the polypeptide chain based upon the side chains of the amino acid residues surrounding the site of cleavage. So there are critical parts here that are highly conserved, but depending on the other residues around them in the active pocket or the active sites, there are different different substrates that they're interacting with. Chymotrypsin is responsible for cleaving peptide bonds following bulky hydrophobic amino acid residues such as tyrosine, tryptophan, phenylalanine, and methionine. All of them are pretty big or containing the phenyl ring, which fit into a snug hydrophobic pocket here. But trypsin prefers lysine or arginine, and elastase prefers alanine, glycine, and valine, which are small amino acids. So based on their shape, structure, and the constituents of those pockets, they have different substrates. The main player in the catalytic mechanism of the serine proteases is the catalytic triad, including the critical serine 195 residue. The triad is located in the active site of the enzyme right in the middle of the, in this case, the chymotrypsin, where catalysis occurs and is preserved in all serine proteases enzymes. And the triad is a coordinated structure consisting of three essential amino acids, which are histidine 57, serine 195, as well as aspartic acid 102, which are located very close to one another near the heart of the enzyme, again in the active site. And these three key amino acids each play an essential role in the cleaving ability of the substrates, which are polypeptides. And in the event of catalysis, an ordered mechanism occurs in which several intermediates are generated. So you can see it in this sort of simplified diagram. The catalysis of the peptide cleavage can be seen as a ping-pong catalysis in which a substrate, which is a polypeptide being cleaved, binds to an enzyme to form the ES complex first. And then a product, especially N-terminal half of the peptide is released there in the complex, and another substrate, water, binds to the enzyme and then releases the another product, which is the C-terminus half of the peptide. Each amino acid in the catalytic triad performs a specific task in this process, which is called the charge relay system. And let's talk more about this system. So the first step of the process would be the substrate or the polypeptide binding into the active site of an enzyme, but this will still have oxyanion hole, which inhibits the interaction between the other residues and the substrate for further process, especially for the cleavage of this enzyme. So these triads are responsible for getting rid of oxyanion hole as well as the actual cleavage that happens on the cis bond. So first, the serine has the hydroxyl group that can act as a nucleophile, attacking the carbonyl carbon of the cis bond of the substrate right here. And a pair of electrons on the histidine nitrogen has the ability to accept the hydrogen from the serine hydroxyl group. So it takes this hydrogen off of the this hydroxyl group. So the oxygen will be more electronegative. And interestingly, this aspartic acid right here in the catalytic triad also makes the hydrogen bonding with this histidine to make this nitrogen more electronegative, so easier to get this hydrogen out from the hydroxyl group of the serine, eventually making it easier for this oxygen atom to attack this carbon. Zymogens are inactive precursors of an enzyme, and they have large inactive structures which have the ability to break apart and change into the smaller activated enzymes. So there are pre-processed enzymes. And think about it. If the digestive enzymes were active when right after the synthesized, then they would immediately start chewing up the organs and tissues and cells that they're synthesized them. So zymogens are basically working as a safety lock. And the difference between the zymogens and the activated enzymes lies in the fact that the active site for catalysis of the zymogens is distorted. So its structure is slightly different than the active one on the active site. As a result, the substrate polypeptide cannot bind effectively, and proteolysis does not occur, but only after the activation of the zymogens, during which the conformation and structure of the zymogen changes and the active site is open. Now the proteolysis can occur, and that is already the mature enzyme. Certain enzymes require cofactors because side chains of the amino acids in the protein provides only a limited range of chemical reactivities and carrier properties. But cofactors broaden the range of enzymes' catalytic properties, so they serve as enzymes' chemical teeth. And there are many different kinds of cofactors can fine-tune or actually completely change the enzyme's activity. So cofactors consist of metal ions and coenzymes, which are typically loosely bound and cycle on and off or in and out of the enzyme. And also, coenzymes have co-substrates and prosthetic groups, which are typically tightly bound organic groups, and oftentimes they have covalent bonding to the enzyme. And holoenzyme, which is an active enzyme form, is a complex of apoenzyme, an inactive enzyme, and the cofactor or cofactors. Coenzymes are changed chemically during the reaction and must be regenerated. So prosthetic groups are regenerated in the distinct separate reaction and can be later added onto the enzyme to form the holoenzyme. And the loosely bound coenzymes diffuse out from this enzyme complex, and their regeneration can be carried out on a different protein, and then later on it can be added to form on the apoenzyme to form the holoenzyme. By their source, coenzymes can be classified into metabolite coenzymes and vitamin-derived coenzymes. Metabolite coenzymes are synthesized by common metabolites, including nucleoside triphosphates such as ATP and GTP, but also include the uridine diphosphate glucose and UDP glucose and S-adenosylmethionine. And ATP can donate all of its 3 phosphoryl groups in group transfer reactions to become the adenine, but it gives those phosphoryl groups to the enzymes for their activity. And UDP glucose is a source of glucose for synthesis of glycogen in animals and starch in plants. And S-adenosylmethionine can donate its methyl group in biosynthetic reactions. The other classification is vitamin-derived coenzymes, and vitamins are required for such coenzyme synthesis and must be supplied in the diet. And lack of certain vitamins cause various types of human diseases. And there are 2 categories in vitamins, water-soluble and lipid-soluble vitamins. Water-soluble vitamins include vitamin B and C, which are required daily in diet, and excess amount of such vitamins will be excreted in urine, as opposed to lipid-soluble, such as vitamins A, D, E, and K, should be limited in terms of their intake, and they're going to be stored in fat. Many vitamins are coenzyme precursors and they must be obtained in the diet. Without enough intake of such vitamins will lead to a variety of human diseases. Here I listed a few vitamins and their sources.